Okay, so we ended the lecture on phylogeny. Well, we didn't finish, but we ended right about here. We were talking about homoplasy versus homology, and if you think about these phylogenies, they're all founded on the idea that two things are similar. Right? So if we go back to this, right, we would say, well, species D and E are similar in that they have a football shape. And it turns out on our phylogeny that there was a little branch that had D and E, and they both had a football body, right? So that suggests that this is a homologous trait. There was mutations in the genes that happened here, and this common ancestor had those genes, and therefore D and E inherited those genes. Okay, but what do you do if two organisms share a similarity, but they did not inherit that similarity from a common ancestor? How do you interpret that, or how do you... Basically, it makes these phylogenies very confusing. So this fish example contains a homoplasy, and so let's see how that complicates things. So we're going to start the same, though. We're going to think about the species. We've got A through E, and we have the different characters. So we're going to have A. I don't know. You can probably, some of you are smart enough to sit and look at this like a puzzle and just figure out how the phylogeny is going to go. The rest of us probably want to start with a table. Okay, so we have our table, right? We're going to think about what traits are variable among this group. So if you look at these group of fish, right, you'll see things like, it looks like there's probably 3 traits that are variable among the groups. One is spots. Right, and then the other 2 relate to fins. You've got the dorsal fin on the dorsal, the top side of the animal. Right, we've got dorsal fin. And then we've got the tail fin, or caudal. C-A-U-D-A-L, caudal fin. Right. Those are the traits. We've got 4, so we have 5 animals, so this should be enough to kind of figure out the relationships here. All right, we need a starting point, which is always your outgroup. That's your comparison to determine what is primitive. Here we're telling us ourselves that A is the outgroup, so therefore A gets a 0 for everything. Okay, and then for B through E, we're going to mark 0 or 1 depending on if that character state is primitive or derived for that species, i.e. is that character state the same or different from the outgroup. Okay, so if you start with B, we look at spots. It has spots, outgroup does not, it gets a 1. B, dorsal fin, species B has it, outgroup has it, so it gets a 0. Caudal fin is forked versus the outgroup is straight, so therefore it gets a 1. Right? And then you just proceed through and mark all these things. And it looks like species C is derived for all the traits. Species D is derived for dorsal and spots, and primitive for caudal. And species E is primitive for everything except for the dorsal fin. Species E doesn't have spots, straight, dorsal, but has a caudal. Okay, so we can see I think the table kind of just gives you a visual way to get started. Now, because there's a homoplasy in here, it is a little bit more difficult to just look and see. Hold on one sec. Okay, so kind of forgot what I was talking about, but basically you can look here and see, get you a starting point, right? We can look and we say, well, obviously it doesn't go from A to C because C is derived for everything. But the thing is, with this homoplazy, is a little bit more confusing because it could go from A, right? Has 3 zeros, and then E is actually the one that has the next fewest derived characters. Possibly it could go to B. B and D have the same number of primitive characters. So you could easily get paralyzed without moving forward and knowing where to go. So just start. Just do something. Okay? If you think that there's 2 different ways this could go, try them both. Right? So we're going to start putting A. It's a little small. Yeah, it's too small. Here's A. Okay? And then let's do with E. Let's try E, right? What changes between A and E? Just the dorsal fin. Dorsal. Okay? And then from E, where are we going to go? From E, right? It could go from from E to B or E to D. Let's try E to B. Okay? And what changes between E and B? Well, actually, everything changes from E to B. We got 1, 2, 3 things that change here. And then if we go from B to, say, D, that leaves us C, A, B, C, D, E. If we go from B to D, then this changes, that doesn't change. There's 2 changes there. And then from D to C would be just 1 change, right? I'm not going to write in all the characters in here. I'm just kind of doing this as a back of the envelope. Because just bear with me. Okay. Now, let's try the other way. Let's try it the other way. Let's start with A. Can you see that? Yeah. A. Instead of going to E, let's go to B. Okay, and what changes from A to B? We have the spots, I think that was. Yeah, spots. And I think this was dorsal. And this was the tail or the caudal fin. Okay, so what changes between A to B is the spots and the caudal fin. Okay, now if we go from B, where would it logically go from B? Probably to D. B to D? No, B to C. Okay, because we have just 1 change from B to C. The only thing that changes from B to C is the dorsal fin. Okay, and then from C, probably go to D. So that puts E at the end here. And from C to D, we have the caudal fin change. Caudal. And then from D to E, the thing that changes is spots. Okay. Let me just make sure this works. So you want to check your work. A to B, we have 1, 2 changes, 1, 2 changes. From B to C, we have one change, check. C to D, we have one change. And from D to E, we have one change, just one change. Okay, both of these possible phylogenies do explain the evolution of this group of weird fish, A, B, C, D, and E. However, one of them is the most parsimonious group. One of them is going to be considered the best because we have 1, 2, 3, 4, 5, 6, 7 changes here. 1, 2, 3, 4, 5 changes here. All things being equal, this one is the one that is correct. But you'll see on here there is a homoplasy. Okay, because what happens, we end up at the end, it tells us that E is actually the most derived group. Well, D and E are here. They were the most recently evolved group. But there are things that E has, like for example, E doesn't have spots. Okay? So E has lost its spots here. But back here, the common ancestor of B and everybody else gained spots. So we gained spots and we lost spots. So E looks like A. That's a homoplasious character. Okay? And this actually, homoplasies are very common among animals. And they make phylogenies a little bit more confusing. Okay, finally. And actually, this one is the most realistic type of phylogeny. And I'm going to get you started with this, and then I want you to do this on your own and bring it to class on Thursday. Okay? In this phylogeny, we have same A through E. Okay? And we have the characters here, though, are not about the shape, the size, bone structure. You could really, any character you can code to make a phylogeny. You can use behaviors, you can use bones, you can use colors, right? But most of the time what people are using is the DNA sequences, the similarity and homologous DNA sequences. Okay, so we're assuming on this example, we have these, well, I can't really see. Oh yeah, we have 4 DNA sequences. And we're assuming that this base is on the same exact place on the chromosome for each of these species. That is to say, it's not, we're taking the DNA sequence of chromosome 3 for this species and chromosome 4 for this species. We're going to the same exact spot on the same chromosome, and we're looking at the DNA bases at that exact area, right? So what we find is that there are mutations and that there are variable places on the chromosomes. And the numbers at the top, 3, 6, 9, 12, 15, 18, those are codons, right? So 3 is the 3rd position in that gene. For species A, it is a C. Species B, it's a T. C for C. C for D. T for E. Okay? So first thing you have to do is find a position that is variable, right? Say the first base is A, adenine, for all 5 species. That's not informative. It's not going to really help you to differentiate among this group. So you're going to look for and find the bases, the base pair positions that are different. Okay? So we see 1 and 2 are the same. Base pair 3, that's variable. Base pair 4, is the same. 5